Okay, hi RLA kids and those of you that missed the lab in school. So you're going to click in this Thursday and Friday folder. This schedule will change a little bit. I just can't change it until tomorrow and I got to make the video today. So if you're an RLA student or you missed the lab in person, you need to go to this folder, Starch Iodine Lab. This video will be in the folder, so hopefully you find it. Okay, and you're going to open this lab instructions and it looks like this. You need to read through them. So a cell model is used substituting dialysis tubing for a plasma cell. The movement of chemicals can be observed due to color change. A positive indicator of a starch and iodine results in a blue and black color. So essentially what we're doing, you can read through those. Essentially what we are doing today is you have one beaker full of water and iodine. And then you have this dialysis tube, kind of looks like a paper bag, like a Tootsie Roll. Oops, kind of like this. This is my plastic bag from lunch, but it looks like this. And on the inside, you have starch and water mixture, okay? So you have this Tootsie Roll starch and water mixture in the dialysis tubing, and you are going to drop it into the iodine and water beaker, okay? Now, you can watch this virtual lab for the setup and to see what happens. So this allows you, so you see this is the, oops, let me rewind it. So this is like the Tootsie Roll. Inside of here is starch and water. That is the dialysis tube. And you drop it in to this iodine beaker, okay? And then you wait. You can watch this video, which you need to, for observations. The result of iodine mixing with starch results in a blue-black color, okay? So keep your eye out for that. Now, as you go, you need to fill out this lab. Where do you find it? Right here, Plasma Membrane Model Lab. When you open it up, it'll look like this. It'll have questions. So you need to record the color of starch inside the tube before, so right when you drop it in, color of the iodine outside of the tubing, so this color before, and then after you watch the video, okay? Which of the following items is used as a model for the cell membrane in the lab? The membrane of the model, or the cell membrane is modeled by the dialysis tubing, right? So that tubing, this tubing is porous. It has little holes in it. And what you're going to determine today is, can starch leave the tube and enter the beaker, or does iodine enter the tube? One of those molecules is too big. It cannot fit through those pores. One of them is small enough. This whole representation represents diffusion, okay? Moving from a high concentration to a low concentration. It's like when you have a helium balloon, a balloon full of helium, and you pop it, that helium doesn't just stay there, it diffuses, it moves around the room. Why? Because it wants to be as spread out and equally spread out as possible, okay? So when starch reacts with iodine, remember you can see that blue color. Here you need to label the different parts. If you don't know how to do this, you can watch the video from Wednesday and it will help you. Okay, this line right here represents your cell membrane, which is your dialysis tubing. Some molecules can cross it, other molecules can't. Which ones? And then I ask you some post questions. Did iodine move into the tubing? How do you know? Did starch move out of the tubing? How do you know? Well, how would you know? So if iodine moved into the dialysis tube, the tube's going to be purple. If starch has the ability to move out of the tube, the entire beaker is going to be purple. Understand what I'm saying? This is called selective permeability. All right. So diffusion, let's look at this real quick. So diffusion is moving of areas of high concentration to areas of low concentration. So here we have these molecules on the left-hand side of very high concentration. Are they going to stay there, shift to the right, or shift to the left? Well, in this one, they're going to shift to the right, right? Because it wants to be spread out. It wants all those molecules to be spread out in that entire beaker, not just clumped on the left-hand side. All right, so this is what you do today, this lab, okay? You have this video to help you. You need to watch this video that comes from here for your observations, and you need to submit it by 11:59 p.m. Okay, let me know if you have questions.